Great. Good morning, everyone. I hope you had a fantastic first day and a good night yesterday. Um, what I realized is that I had a pretty, a good copy and paste mistake in duplicating slides. Of course, NASA figured out a lot of different things about life and planets and all this kind of stuff, but a lot of data is not from NASA. That was from the first slide, and you duplicate it, and then you make your new slides, and you realize in the end, and then you have this little mistake. But anyway, so yesterday we went through very basic concepts in biology, learning a little bit about what is a cell, right? And what is a cell made of. Um, and today I'm just going to continue a little bit in the basic idea, and I just want to also touch a little bit on metabolism, what Sunil did, and just build up a little bit on these concepts of energetics and metabolism. But then also, since my lab is interested in the energetics of development and developmental biology, I will also give you a brief introduction into, I think, a fascinating field of developmental biology. And this field that also has become now a little bit of a playground for quantitative biology and physicists trying to figure out how forces and cellular mechanics shape and developing organisms. But for that, I just, in the end, hopefully give you a brief introduction into developmental biology. And one beauty is that, we learned something about a basic prokaryotic or eukaryotic cell, but if you look around in organisms, we have a huge variety and diversity in cell morphologies, right? So in principle, you can use a stem cell, and if you know how you change fate and differentiate the stem cell, you can, make a lot of different kind of cells in a body. For example, immune cell, an epithelial cell, a nervous cell, or blood cell. And how do we know this, right? Like, how do we figure it out over the last century of science? What are different cell types, right? Like, we had model organisms, and I just briefly want to give you a little bit of an idea what kind of model organisms we in biology use to figure out, what are different cells, what are different cell types, right? And there are a lot of people working on the development or the physiology and the biology of plants. And there's one model plant which is used, which is Arabidopsis thaliana. And on the other hand, we have also an invertebrate, like it's a little tiny worm called C. elegans. And it's, it's roughly a millimeter in size. It can either reproduce sexually or asexually. Then we have the famous fruit fly, Drosophila melanogaster, where a lot of principles in developmental biology, how you pattern a developing embryo, have been determined. And then there's a vertebrate known as this frog, Xenopus levis or Xenopus tropicalis, and this is a male and a female and a male. And my favorite model system, or the model system we are using, is also like a vertebrate. This is this fish, what we call zebrafish, because it has these beautiful little shiny stripes, and it's called in Daniario. And then a lot of translational or more applied science have been actually done in the mouse. And the beauty, why did people use mouse, is because if you compare a human and a mouse, they have very, very, very similar genetics and very similar embryonic development. Although in the end, we are of course very different between a mouse and a human. But before I get into developmental biology, I just want to talk a little bit about what we did yesterday. We learned a lot about size scales, right? We learned how big is a classical prokaryote, how big is a eukaryotic cell, how big are different organelles, how, what is the size or the volume of a prokaryote or eukaryote. But of course, all these processes or biology is active and happens in time, right? Like if you think about development, it has the trajectory of time. So at one point, it would be also good today to talk a little bit about, for you to get a feeling on what timescales do different biological processes happen. And if I'm going to just ask maybe not the developmental biologists in the room, but how long do you think does the development of a fly take? A few days? So in the timescales of days, and everyone agrees? Exactly. So it's in the timescales of days. What I find remarkable, you go from a single cell and the egg within 1 day to a larval stage, right? So within 1 day, you take a single cell, you build up roughly about 50,000 cells, and you have dimensions, right? You have a front, you have a back, you have a top, you have a bottom, you have a left and a right, and you have organs, right? You have a gut, you have a brain, and all this developed from a single cell within 1 day into a full body plan. I find this amazing. And then, of course, the fly uses about 3 days to go from this tiny little maggot, and this tiny little maggot is growing massively, about 120-fold in size and in weight over 3 days before it's going to undergo metamorphosis, where this larva is now going to form a pupa, and then all these organs, which you have previously built, are going to get remodeled, right? Some of these organs are going to get completely destroyed, and some organs going to get completely made new, and you're going to form what in the end then looks like a fly. And that process takes about 9 days. It's actually very, very, very fast for a developing system. But then if you think about how fast do you think these early divisions are, right? Like in the beginning, you need to make a lot of more cells, right? So how long do you think a cell division in a developing fly embryo takes? Hours, days, minutes, seconds? Minutes to hours? Okay. So the fly is actually very special because it has one of the fastest division cycles, because you not necessarily in the beginning make more cells, but we can go into this in a little bit more detail. So basically you have this big cell and you have a single nuclei, and then within, about 10 minutes, every 10 minutes, you make another nuclei. Ba-bum, 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 ba-bum. And eventually you end up with a big cell with many nuclei in it. And then you're actually going to form the membranes of these nuclei. And this is what we call a syncytium, right? It's a single cell with a lot of different nuclei in it. And eventually you're going to form, during a process called cellularization, you're going to form the cells, and then you're going to remodel these cells. And this process roughly takes about, every 9 minutes. Is one of the fastest, DNA replication cycles out there, at least in an animal. And then on the other hand, if we compare this to bacterial cell division, where you now, of course, double your mass, you have to separate these two cells. And this one also happens on the scales of minutes, but more on the scales of hours. You can have, like a division time of roughly about 30 minutes. And, how do we figure this out, right? And basically, this is where microscopy sometimes comes in, right? Like you can, for example, take an E. coli cell under a microscope, and you can watch the E. coli cell doubling over time, right? And if you have enough time resolution in your frames, you can segment these E. coli cells and then just look at the area of the pixels over time and see when this is doubling. And then you can deduct, for example, your doubling time of an E. coli. On the other hand, this E. coli is also moving, right? And the cell movements also now for a bacterial cell now, we go from the minute time scales into the second time scales, right? So it's roughly like, very randomly movement. There's also directed movement, chemotaxis in E. coli. But generally, cell movements happens on the scales of seconds, at least for bacteria. And then if you think about intracellular diffusion, we can think about, we have a cytoplasmic protein. And then the question is, how long does it take for a cytoplasmic protein, for example, to traverse a cell? based on diffusion. So you can then think about what is the diffusion coefficient of this molecule. Roughly, it's about 10 micrometers squared per second. And you can do this in 3D. And then you think, okay, this molecule on a size scale of an E. coli takes about 10 milliseconds to diffuse around a cell. Now, if we take the same protein and put it in a bigger cell, for example, like a mammalian HeLa cell, which has a length scale roughly about 20 microns, this molecule now takes about 10 seconds to go from one side to the other. Right? And now, if you think about the longest axon we have going from your hip to your toe, and if you want to transport a molecule from the cell body where it's made down to the tip of your axon, and you think, I'm just going to do it passively via diffusion, and you can calculate how long this is going to take. You know, it takes days or even weeks, depending on how long your axon is. So that's the reason why, for example, molecular transport in your axons is active, right? You have your molecule transported via the cytoskeleton, what we heard yesterday from Jingbo, usually microtubules, where you have cargo transported actively down your axon, because diffusion would be just way too slow to bring this molecule. On the other hand, we talked about what is transcription and translation, or protein synthesis and gene expression. And these guys usually happens on timescales of roughly seconds. Right? So if we look, what is faster, transcription, translation, we see that they are roughly operating with similar rates. So making your molecule, transcribing your message from the DNA into RNA is also kinetically coupled to translating this message into protein. So how do we know how fast an RNA polymerase is? So if you say, in my model, I have a parameter, I need to put the speed of an RNA polymerase in there. And then you come to an experimentalist and say, can you design an experiment to get me this number? How would you do it? Do you have any idea how we can do this experimentally? Just anything goes. So if I tell you, design me an experiment to tell me how fast an RNA polymerase is. Can you use the mic just that everyone can hear you?
Hello?
Yeah.
Yeah. So we could use radio-labeled nucleotides,
Mm-hmm.
and the speed at which probably the radio labels are going up, we can sort of get the time scale
Okay.
at which.
Yeah, that's a good idea. It goes in the right direction for sure, right? And then you need to pulse chase. So do you, or
Yeah, so the nucleotides will be available in abundance.
Yeah.
So as and when, like, we have to just trace the graph as to how the labels are being incorporated. So
that's a good idea. I think that's one of the classical experiments you could do. There was another raised hands over there.
I don't know. Yeah, it was just about like measuring the RNA rates into a cell, and then we try to report this number to a number like of RNA polymerase. So
The experiment for RNA is actually very similar. They use the label, right? So for example, now think about the gene structure of the structure, and you have a promoter where a transcription factor is going to bind and going to initiate a transcription of an RNA which you designed. Right? So for example, you can think about designing an RNA molecule which has a sequence which is going to fold in 3D, right? Yesterday we learned that RNA can fold in a 3D structure, and this fold can be used to be bound by a protein, right? So for example, here, this is a favor structure which we call an MS2 stem loop. And we can use this because the MS2 stem loop can bind the MS2 stem loop binding protein, which you can put a fluorescent tag or fluorescent protein on it, like a GFP. Right? And then what you can do is you can put the stem loop either on the left side of the RNA, on the 5' end, or on the 3' end at the end of the gene. And now you take this designed transgene and put it in your cells, and this, I think, is even done in a Drosophila embryo. And then you can follow the appearance of the 5' end, and then you have another construct, the appearance of the 3' end, while using this protein binding to the stem loop. And then you can plot the frequencies of the time, how long it takes for these spots to appear, because you can switch it on and off, this gene, using this promoter. And then you can see the time differences between the onset, or the 5' end appear, like the 5' end appearing spot and the 3' end appearing spot, and you know how long your gene has been. And then you can just take the length of your gene divided by the time difference, and that roughly gives you this idea of about 25 nucleotides. Does that make sense? All right. How would you do it for an RNA, or like for a protein translation? So basically, you have an RNA molecule, and you have the 5' end here, and the 3' end here. So you have a specific sequence over here, where you have bases. Let's just say these bases are complementary to each other. Right? If you then wait in time, what's going to happen is that these complementary bases are going to base pair with each other, right? And going to form it like a 2D or 3D structure, right? That's the 5' and the 3'. So now you're going to have a protein, which we call the MS2 binding protein, where you have another label on it. For example, that's this famous molecule green fluorescent protein. So what's going to happen is that this protein is going to bind this structure. Okay? So now, if you think about it, you have your RNA polymerase transcribing this from the DNA sequence. So first of all, nothing's going to happen. Then you're going to wait some time. And eventually, depending on where your stem loop is, you have transcribed this sequence. And then it's very, very quickly going to fold into the stem loop. Okay? And then you have your binding protein binding it, and then you're going to see that spot. And then you can do this either early in the gene or late in the gene. Okay? Does that make sense? So now I want to know, how fast does the ribosome translate my RNA? How could we do an experiment to figure this out? The problem is that a peptide sequence is going to fold, but it's going to be very, very hard to design an interaction motif on the timescales where translation is happening. However, what people have done is your RNA molecule is going to have a lot of ribosomes initiating, moving around your RNA molecule to translate it. So at one point, what you can do is you can prevent new ribosomes from coming in using a small chemical drug. So now, if you think about it, you have a lot of ribosomes on your RNA molecule. And now, what you prevent or you stall from new ribosomes coming in, okay? And this prevents new ribosomes coming in, but the other ribosomes are going to move, right? So if you now do that in timing, right, like you take samples every time interval after you inhibited or after you stalled the initial ribosome, and then if you have a method to figure out where the ribosome is on your RNA molecule or which position it has, you can do a time course and see how these ribosomes move. And this is what you can do, for example, with ribosome footprinting, where you degrade your RNAs and you sequence this little piece of RNA bound by your ribosome. And if you know your sequence, you can especially tell where it is over time. And then you can plot this position against your delta time intervals. And then from the slope, you can then deduct how fast these ribosomes are moving in your system. Yeah.
I guess this is the modern way of
That's the
measuring.
modern way, yeah.
Is there a classic way? I'm sure people have measured this or at least estimated in a more rough way before this
ribosome footprinting. I think people have done this, but like right now, out of the top of my head, I don't know the exact experiment. But how would I do it with classical experiments? So you can definitely cross-link your RNA to your ribosome. And what you're going to see is, if you put that on a gel, you're going to see it shift in the mass of your ribosomes bound to your RNA. So now, let's see if this is going to work. If you now inhibit translational initiation, your ribosomes is going to fall off. And I don't know if you have enough time resolution to get this position on the gel, but you probably could. Or you do radioactive amino acids. And you have radioactive amino acids on your tRNAs, and then you do scintillation counting as a function of time. And then you would assume that the radioactivity of a single amino acid is proportional to the amount of amino acids you have incorporated. And that from that, the radioactivity then should be proportional to, and then you do that over time, you spike it in, and then you could probably get it. All right, so now if you think about, now we can go even in the millisecond scale. So if you think about, ion channels in your cells or, like electrical signaling, this happens on the scale of milliseconds. And yesterday we heard that metabolism is very fast. And if you think about enzyme catalysis, now we are in the scales of microseconds. Exactly. So an enzyme can actually be very, very, very fast. And there's just a little bit of techniques, right? Like if you think about it, if we have a time scale, for example, milliseconds to hours, this is in the time scales where you can use microscopy to directly observe, if you have a proper designed experiment, to directly observe these time scales. So for example, this is a macrophage moving around in a blood sample trying to catch a bacteria. And there's a famous movie, which I didn't find, but this is the little bacteria over here, and the macrophage is chasing it. And if you want to know, for example, how fast this macrophage is moving, of course, this happens on time scales or crawling. On the time scales of milliseconds to hours, you can use microscopy to observe this. There's another way, if you have milliseconds to years, another way you can do fixed time points, right? Like, for example, here, if you want to, for example, observe what is the doubling time of my E. coli population, I can, for example, do classical experiment. I want to do a growth curve of my E. coli every hour. I take a sample of my culture, and I measure the optical density or the absorbance at 600 nanometers. And then I plot these optical densities against time, and from that, I can then deduct my timescales of growth. Or similarly, how did we figure out how long does it take for a molecule to go from the cell body of a neuron down to the tip of an axon? Is you used exactly what I mentioned to Simone earlier on. You can use radioactive labeled amino acids, and you pulse the cells with radioactive amino acids. And what's going to happen then is these amino acids going to get charged in the tRNA, as we learned yesterday, and then incorporated into newly translated protein. And a lot of proteins, like the proteins are made here, but the proteins which need to go there will now have this amino acid. And then we can use radiography just to see where this radioactivity is in these neurons and follow over time in a population, for example, in one cell, where is my radioactive amino acid? And we assume that this is in a protein, and then you can see it's made here and initially then transported down the axon. And then you can figure out how long does it take for this protein to get there. Or similarly, you can then, for example, also look, if you want to know how strong is a certain gene expressed, you can just look at the accumulation. For example, if you express a GFP in a cell, you can see how long, like how much is it expressed. You can follow the GFP fluorescent over time using microscopy. And this is just a summary. We also saw that yesterday, just about the time scales we're talking about. And if we go top down, right, like we have the generation times, in roughly in minutes. And then we can even go down to, the average catalytic protein turnover, to ion channels, to electron transfers and so on. That's just for you to get a little bit of an idea, how fast do biological processes. Are there any questions so far? Good. Then there's an important number we sometimes need, which is the turnover number. And then if you think about, if we have a molecule we are interested in with a certain pool size P, we assume it is at steady state, meaning that the formation rate and the usage rate are the same. We can define a flow rate, which is just the formation of the usage rate, called V. And if we then want to know what the turnover time is, we have our pool size divided by V, which now has the units of seconds. That's the turnover time, for example, of, a metabolite or ATP or something else. And if we now just have a look, what are just general turnover times in energy transformation biology, you can now see that they're roughly on the orders of seconds. So if you think about, how fast are going to my ATP pool turn over, right? Or for example, how fast going to turn my, like whatever, some intermediate in your glycolysis or something else turn over, a good initial guess is roughly on the order of. Also, not only metabolites have a turnover time or like a half-life time, it's different, but for example, if you think about, how long are going to be my RNA molecule around, be able to make protein, it has a half-life time. And if we compare different species, you can see that the half-life time of RNAs are roughly on the order of minutes to hours. So if you transcribe your gene and you make this disposable pool of messenger RNA, all these little molecules going to turn over roughly on the scales of minutes and hours.
Quick question, which is just a clarification. So here, when you talk about turnover, you're talking about degradation, right?
Yeah, active, yeah.
But in terms of ATP, you're talking about how is the turnover time defined?
This is the turnover time, right? The turnover time is defined if you're at a steady state, right?
Yeah.
Then you say that the flow rate through the system is either the outflow or the formation rate, right?
But this is the formation of ATP or is the phosphorylation?
This is the phosphorylation. Just like
Phosphorylation,
not
yeah.
the not the de novo synthesis. That's of course going to take much longer.
Okay.
Right.
Okay.
Yeah.
Just that was the clarification.
Okay. Yeah.
Type of mRNA, is it that mRNA is once made and then it's translated and then it just degrades over time, or is it, are these times like actively controlled?
So like an RNA can be used multiple times, right? And there's active degradation of RNA. So we have these RNases in cells, which gonna actively degrade your RNAs. And there are some RNAs which are more stable than others, and you have certain sequence elements which make them more or less stable. Of course, if you ever work with RNA in a lab, you may realize that they degrade faster than you want because you somehow been either not sterile enough and put some RNases in there, but they're also naturally sometimes unstable molecules because you have this little OH group which can chemically and nucleophilically attack and cleave your RNAs. Yeah. Yes. So there is, so normally like an RNA breaks down into nucleotides. You can either polynucleotides or even single nucleotides. But then if you look at the structure, right, if we remember when we made an RNA, we took like an NTP, like a triphosphate, and you have then it's hydrolyzed into pyrophosphate, and you have this monophosphate bond between the two nucleotides. So what's going to happen if you cleave it? You're going to end up with a mononucleotide molecule. So it's exactly, and then you can either phosphorylate it again, but you need to put energy in it. And there's also other ways you can reuse. It's called the salvage pathway, but I think we don't want to go too much into that detail. Yeah, you can. Like a ribosome can fall off, and then you're going to have some what we call a truncated protein. Right? So, and that is usually then also detected by cells and then actively degraded, because you don't want that to accumulate, because it could do something you don't want. All right. Similarly, we have half-life times of proteins, right? And that is on the scales of hours, and sometimes even days. So this is just like an average half-life time of proteins in the yeast cell, and it's roughly on the order of an hour. But also in human cells, you can already see there's some interesting trajectory in terms of half-life times and timescales happening in a single prokaryote and a single eukaryote. You're like yeast. We go back, there's a single prokaryote, a single eukaryote, and a mammalian cell. You can already see that these timescales are very different. Right? And similarly, for the protein half-life time between a single eukaryotic yeast cell and a single human cell, there's a very different timescale also in terms of how fast they divide. And I don't know, Mike Lynch might go a little bit into the idea of metabolic scaling with size and thinking about how that might be related to speed or time and biology. But anyway, right, we can tell time, right? I don't know if you know, but we have a clock, an inner clock, which is called the circadian time, circadian rhythm. And our physiology is different depending on the time of the day, right? So for example, in the night, our blood pressure changes, our metabolism changes, and so on and so forth. So how do you think a cell or an organism can tell time? Thinking about molecular mechanisms. How does a cell know what time it is? Okay, that's one way. So, and then what, right? Okay, so you have a reaction determining with a rate. You need to do something else in order to be able to tell time. Right? Exactly. So oscillators, right? You also have same thing? Okay. So there's this famous way of biological oscillators, which, has been discovered for the circadian clock. So for example, if you look at a protein, this is a algae protein called Chi-C. And somehow, if you take this protein out of your algae at different times, you can see that this protein is going to behave on what we call, when we separate it by size, behave differently. And we know that this behavior on this gel is dependent on this phosphorylation of this protein called Chi-C. And here you can see that over time, this Chi-C gets phosphorylated, then dephosphorylated, phosphorylated, dephosphorylated, phosphorylated, and dephosphorylated. And on the one hand, you can then plot these intensities and then do the ratio, and you can really see, nice oscillations in this phosphorylation, dephosphorylation state of this protein. And it nicely oscillates between these two states. And this is what we know as the circadian clock or the circadian oscillator. And it has a period, it has a phase, and this is how, at least on this time scale of 24 hours, if this phosphorylated protein has an activity, we now can then change gene expression, so on and so forth, in order to tell time.
So when you were talking about the half-life of the protein, which is around 40 hours,
Mm.
so that is starting from synthesis, or it also refers to the phosphorylation and the dephosphorylation of the protein? So what would be the time scale?
that refers to synthesis and degradation. These phosphorylation, dephosphorylations are usually faster,
What would be the time scale of
roughly seconds as well. This is an enzyme which uses ATP in order to put the phosphor group on their protein. It's roughly on the time scales of seconds. Exactly. So, and then you had another question.
Yeah, so I kind of wanted to ask, like, how long, I mean, how early did the circadian rhythm evolve? Like, do bacteria have
Sorry,
it?
I didn't hear you.
Do bacteria also have circadian rhythm, or is it just something that's found in higher organisms?
I don't know. Does anyone know whether bacteria have a circadian? I think a lot of cyanobacteria have it, right? Of course, the ones which do photosynthesis. This is cyanobacteria or algae, right? Yeah, I think this data is from cyanobacteria. That's the cyanobacteria, yeah. Yeah. Exactly. So this phenomena of biochemical oscillators is very widespread. So, for example, the eclosion of Drosophila is also, eclosions means like, getting out of your pupa shell is regulated by an oscillator. You have the cyanobacteria we just talked about. Birds, melatonin levels are oscillating. And the general structure of these regulatory systems, you have an environmental input, which leads to the expression of a positive element, leads to the expression of clock genes, which target is also negative element. So you have a negative feedback loop and a degradation, which then leads to the oscillation of your protein or even your activity.
When you showed the image of the gel, there was no production of the proteins, right?
There is no production. This is just a made protein in a test tube.
So it's stable for more than
This is
2
a very
days?
stable protein.
Okay.
And it's also outside of the cell, so meaning you don't have all the machinery which actively degrades your protein. This is really in a test tube.
So if and that this is true for most proteins, if they're
Outside of the
cell,
test tube, they're actually very stable, usually if you purified them well.
Okay.
Circadian rhythm is calibrated? Is it by sunlight usually?
By sunlight, usually.
Mostly by sunlight.
Yeah. So it's entrained by sunlight. So we in our neurons, we have this central clock in our brain, the circadian clock, and then it entrains all the other clocks in our body. You want to say something? Yeah, sure, please. Yeah.
The circadian is the 24-hour, the light day cycle, but you have far more frequently, you have ultradian oscillators of different time scales in biology. And a lot of them are completely metabolic oscillations. So the whole oscillator is a metabolism-driven oscillator. Like yeast, for example, have an ultradian short oscillation, entirely driven by a metabolic cycle. So you'll go through one round of glycolysis, TCA, come back, oxidative, reductive. And those are far more common in biology. This is, of course, the most kind of visible,
it's a nice example.
dramatic one.
Yeah.
But you have very common short oscillators. They're all driven by a circuit. Sometimes it's very simple like this. Sometimes it's a little more complicated, but you have a nice Feed forward, feed back, and you'll get a nice oscillator. And it could be driven completely by energetics or redox or whatever else.
I wanted to follow up on that. So like these metabolic oscillators, like what purpose do they have again, like for timekeeping or something? Like
Actually mentioned this in my last talk on Friday, so maybe we'll hold off till then. I'll give you an example of a completely metabolic driven oscillator.
Known as the famous yeast metabolic cycle. That's a cool example. Really nice. And our circadian rhythm, or like the principles, have been figured out. And I don't want to go into details, but this is how it looks like. Right? Like, this is the central clock wiring of that system. But we have another famous cycle, which is the cell cycle. Right? And if you think about the classical mammalian cell cycle, what is usually happening is your mass is linearly increasing over the course of your cell cycle, and eventually you have cell division, and you halve. You double your mass, you halve. Double your DNA, half, and it's nicely oscillated. Right? And if we now just go in this basic idea of the cell cycle, we have different cell cycle phases. Right? So for example, if you have your daughter cell, and eventually you grow in mass during a phase, what we call the gap phase 1 or G phase 1, and then you enter the phase of DNA replication, so you're going to double your genome. You're going to enter another phase called gap phase 2, and then you go through mitosis and separating your cell cytokinesis, and then you have completed one cell cycle. And so it's this periodic repetition of G1S, G2M, G1S, G2M. And generally, if you just look at timescales of these different phases, you can see that, for example, in fish and yeast, G1 takes the longest time, like G2 takes the longest time, and in a human cell, it's roughly G1 and S phase are the longest phase. And here's your favorite Drosophila embryo, right, where we don't have growth in a way, because we don't make double in mass, we only have cellularization. And you can really see that they now lack these G1 and G2 phases and rapidly repeat M and S, M and S, M and S. And that is very reminiscent of early development of basically most species going through these rapid cell cycles, which are minimal cell cycle of S and M phase, or very, very short G phase. And there's also machinery behind it. It's the classical, what we call the cell cycle oscillator. And what you can see here is an interface. We have this protein slowly accumulating, which we call a cyclin, because it cycles. Right? This is how it was discovered. It cycles in its abundance, and it's slowly increasing, 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 increasing. And this protein is going to bind an enzyme, or like a protein which we call a kinase. Kinases are proteins which phosphorylate targets, and they use ATP or GTP sometimes in order to transfer this phosphate on your protein. Similarly to and this activity depends on whether it's bound to this protein called cyclin. And when the cyclin is accumulating, eventually it's going to reach a threshold, and the cyclin-dependent kinase becomes active. And then the cyclin-dependent kinase can initiate the transition between interphase and mitosis. And then it's actively degraded. And this has been figured out similarly, classically, now with classical biochemistry, right? So here you use a radioactively labeled methionine as an amino acid. And you can see, how big your cyclin is. And you can see that over time, this amino acid is incorporated in your cyclin, and then the cyclin is accumulating and then degraded. And this is a radioactive assay where you can use radioactively labeled ATP, and your phosphate is radioactively labeled. And then you can see, how is this radioactively phosphate transferred to other proteins. And you can see now that this accumulation of cyclins is now followed by this very sharp peak in this activity of this kinase. And this is basically very, very simplified. We can design a minimal oscillator. Just theoretically, you have molecule A, which makes molecule B, which has a negative feedback loop. We have an outer catalytical positive feedback loop, and this will oscillate in the activity of generally. And then this is your minimal cell cycle oscillator, your embryonic cell cycle, which always goes M and S phase, M and S phase. You can design a minimal oscillator, which is called this MPF, it's mitosis promoting factor, where you have an intrinsic positive feedback loop, and then it produces a protein called the APC complex, which leads to the degradation of this protein, and you have a negative feedback loop, and this time delayed, which then causes this mitosis promoting factor to us. And this is the minimal description of this circadian oscillator. Of course, if you now look at mammalian, like a yeast cell cycle oscillator, it's a bit more complicated than that. But you can coarse grain it basically down to something like this. But this is how we now know the feedback loop structure and the biochemistry of the cell cycle in a yeast cell. Just as there's, I think, decades of biochemistry in this plot. But this is how we kind of slowly figured out how this works. And of course, the cell cycle oscillator then is going to introduce changes in your gene expression. And now with modern sequencing technologies, it's going to either repress or activate certain genes, and then helps to go through these different cell cycle phases, doing different cellular processes. And we can sequence, and you can really see that a lot of genes or gene clusters are a function of time, or the function of the cell cycle oscillator. So you can see that a lot of genes are introduced at different times of the cell cycle progression, and a lot of genes are repressed or downregulated during cell cycle expression. If we now look, right, in our human body, we turn over quite a bit. So all, our skin cells, our gut cells, our liver cells, they all turn over. A lot of different cells in our body turn over. So we have still a lot of active, cell cycles in our body, although a lot of cells are also post-mitotic. Right? One reason why, if you received chemotherapy, or if you, during cancer patients, going to lose their hairs and have a lot of problems, is because we also, if you want to block the cell cycle of your cancer cell, you usually sometimes also block the cell cycle of other cells in your body, which you don't want to target. It just gives you a little bit of an idea what the turnover is. And it's interesting, I don't know if you knew that, but humans, the germ cells in humans, or female gametes, are present when you're born as a baby and you don't make new. Right? They don't turn over. So all females are born with their oocytes already present, or with the germ cells present. Yeah. Lens cells is your cells in the eye lens, exactly. They're also present there. And of course, now if we look at the time scales, for example, of establishing patterns in the Drosophila and the fly embryo, this is all down to signaling systems and gene expression, making these beautiful stripes of tissues, and we will get that maybe tomorrow. But on the one hand, right, if we now go back and say, like, molecular genetics, biochemistry, cell biology gave us a really good list of genes and proteins. But on the one hand, now the challenge is, how do we turn this into, this information into understanding and quantitative understanding? And I think this is why we're all here, is now we're going to use a lot of more quantitative tools in order to turn these lists of genes and proteins into understanding. But now I just want to come back what Neil introduced yesterday and talk a little bit more about metabolism. And we already did that yesterday in the introduction, that we are open chemical systems far from equilibrium, and we have to take energy and matter from the environment. And If you think about it, right, like we all burn sugars, and one classical description of metabolism is that you do this via stepwise oxidation, right? Do you think what helps us to do stepwise oxidation of the glucose compared to just burning it in one step? Why do you think this is helpful to do it step by step? I could just burn glucose, just from going from glucose to CO2 in one step. Kabam. That's one way. What happens if I burn? That's the right direction, right? Like what happens if I just light on glucose or light on wood, right? What happens if I put a spark? Exactly. So you're going to generate a lot of heat, right? And you're going to generate a lot of energy in one step, right? Like you're going to light on a fire, literally. What happens if you burn so much heat? Right, you're going to burn, right? Like your protein's going to degrade and all this kind of stuff. So you don't want to release all that energy in your sugars or in your wood in one step because it's basically lost as heat. So what you want is exactly to build chemical reactions where you do that, you harness that energy in a stepwise and also in a magnitude that the cell, you can use this energy to do useful things. Right, and this is where our beautiful macromolecules come in to do this beautiful stepwise oxidation of energy, right? Because what Sunil already introduced yesterday is that these macromolecules or proteins or enzymes can form these chemical reaction networks. And there are two broad classifications of metabolic reaction networks. One is called catabolic reactions, so breaking down stuff. This is what it means, catabolism. So it's basically catabolism is harnessing or breaking down your nutrients, your sugars, and transforming them into energy currencies the cell can use, such as ATP or reducing. On the other hand, there are reactions which we called anabolic reactions. And I think you heard about anabolic steroids, right? They make you grow, they make your muscles grow, they make you strong, maybe, probably not. And these are the reactions which then build up macromolecules or the precursors for macromolecules. And then together, these catabolic and anabolic reactions form something which we call cellular metabolism. Right, and we, all these enzymes form these, this is what Sunil introduced yesterday, form this cascade of chemical reactions. And he also introduced that what is true for the thermodynamics of the free energy change of a single reaction also has to be true for the whole cascade or the whole metabolic network. So for example, if you have a favorable reaction going from B to C, the whole cascade going from A to F also has to be favorable. And of course, you can have unfavorable reactions in this network, which you then can either, pull, so you have, downstream much more favorable reactions, so you can pull the reaction forward, but you can also push the reaction forward. So you have a very favorable reaction, and if you have a reaction here at equilibrium, right, you're going to add more substrate, and it's want to relax to equilibrium, so you're automatically going to produce more product. So this is push and pull. And then conceptually, right, this is how we understand metabolism nowadays, right? Like it's much more complicated than this, but I think there's a beauty in this, in the chemistry of metabolism, but we're not going to go there. But conceptually, you can think of that your catabolic pathways going to transform energy in your sugars into useful energies, for example, ATP. However, we all know that in order for a catabolic reaction to be favorable, the whole catabolic pathway has to have a negative free energy. meaning that it is dissipative, right? So already not using ATP, but even making energy currencies in a cell, you're gonna lose some of that free energy to the environment. And some of that free energy which you had in your nutrients is gonna be conserved in the free energy of your energy currencies. And then on the other hand, we can then use these free energies to drive unfavorable reactions. And there's a lot of anabolic reactions which are unfavorable.
Again, I'm going to ask a question just to clarify. So are you saying that anabolic pathways do not dissipate heat?
No, they do as well. Like some of them do, right?
Is it much less or?
No, it's in a similar order, right? Like depending, for
Well,
example, if you use ATP to drive an anabolic reaction. So imagine you have an anabolic reaction with a delta G 0 or delta G of roughly plus 5 kilojoules per mole. And then you're going to use an ATP, which depending on the physiological condition, let's say gives you 30 kilojoules per mole. So you drive the delta is 25, right? But the 25 is also lost to the environment, either in form of heat or in the form of entropy. So both of them dissipate. And so you can just, conceptually think it's the same, it's basically the same plot. You have your food molecule, you do catabolism, you have an oxidized food molecule, you produce an active energy carrier molecule, which you then can couple to unfavorable reactions. And this is more of the mechanical analog, right? Like, so you can have a rock falling down, and once it hit the ground, all this kinetic energy is going to convert it into heat. But now, think about this is your theoretical energy you can get from your sugar molecule. And now, what you're going to do is you're going to couple it to energy currencies. In this case, it's going to hit this wheel. This wheel is going to pull up or make ATP from ADP and pull up this bucket of water. Although you cannot take all that energy from that kinetic energy, and some of them are going to fall down and still produce heat. Right? And now you're going to use this bucket to drive a hydraulic machine to do some useful work. But of course, that's not 100% efficient. That's a little bit of the mechanical analog of this anabolic catabolic coupling. And of course, one of this energy carrying molecule is what we already had, is ATP. You have your phosphoanhydride bonds with these 3 phosphates over here, and they contain a lot of energy, and you can hydrolyze them to an ADP, an inorganic phosphate, or even further hydrolyze. And
say that when the graph which had food going to macromolecules, that macromolecules have a lower Gibbs energy than food, or am I thinking about this wrong?
So like the Gibbs energy of formation, you mean?
Mm-hmm.
So what is the macromolecules? The Gibbs energy of formation of glucose is roughly 900 kilojoules per mole. Of course, if you oxidize it, it gives you about 3.5 megajoules. And then the Gibbs energy of formation of biomass, so you can take these chops and estimate. And I think my friend Pablo is currently calculating it. Yeah, exactly. But he will go through it, but it's roughly the Gibbs energy of formation of biomass is roughly 80 kilojoules. That's less. So I don't know, the Gibbs energy of formation is actually a delta G, because it's the free energy of the reaction making your molecule form elements. Right? And it's very different. This is what you plug into your chemical reaction in order to calculate the free energy change of your chemical reaction. So if we now want to know how much energy do I get from ATP hydrolysis? And the concept of free energy was also yesterday introduced, right? We can write down this chemical reaction, right? We have our ATP molecule. And we have water, which is going to hydrolyze or take one of these phosphates away from a phosphoanhydride bond and form this ADP molecule in PI. Right? So then we can take the formula, right? We can calculate the free energy change of that reaction. And of course, at one point, it's related to the equilibrium constant, what we had yesterday. And this is what we call this delta G naught prime. But this is at conditions, at standard conditions, which are very unphysiological, right? So all your reactants are 1 molar, and water is 55 molar by definition. So you can calculate how much of free energy we would get. You can calculate the equilibrium constant and plug it into the delta G, and you roughly arrive at equilibrium, related to the equilibrium, of about 30 to 40 kilojoules or 20 kT. This is because, exactly. However, if we now go into a cell, the concentrations of ATP, ADP, PI are going to be very different from the standard conditions. And we know that, of course, the overall free energy change of a reaction is related to the equilibrium, but then is also a function of the concentrations in your cell. Right? So if you then calculate how much free energy you would get under physiological conditions, we know that ATP to ADP is roughly 10 to 1. We can see that we now usually get a little bit more free energy from ATP hydrolysis under physiological conditions than at equilibrium conditions. No. Well, it's like the equilibrium will be roughly the same. And then it depends on your reaction, like your physiological concentrations of GTP to GDP. Because the molecule is not so much different, right? And most of the business end of this molecule is in here, right? For the free energy in its phosphoanhydride bonds. And whether you have a different base over here, so GTP and ATP is going to differ in this molecule over here. This is the question why is always so hard to answer. And everyone, like I had this conversation yesterday, and everyone thinks that evolution optimized it, but who knows, right? Maybe it has been just around. It was the first molecule, and it just gave you enough energy to do stuff. But why ATP, right? Like, CTP is used, TTP is sometimes used, although much less. And then GTP is used for the GTPases. There's, I think, no biochemical argument or thermodynamic argument why would you use the other over the other thing. But these why questions are always very hard.
So again, what's the typical concentration of ATP in the cell at a given time?
So it's in the millimolar, lower millimolar range. So it's roughly 1 to 10 millimolar. And then ADP is roughly in the high micromolar, like 800, 900 micromolar range. And it's also millimolar is very abundant. So that's another thing, it's a lot. Of course, what is always important is that on the one hand, the free energy is of course related to the equilibrium constant, but it's always dependent on the physiological conditions or your concentrations or activity coefficients, depending how deep you want to go in your cell. Right? And this is what we call this ATP to ADP ratio, since it's going to change how much free energy you're going to get from hydrolysis event. This is what we call an energy charge ratio of a cell, right? And there's an important point is, would you say, based if you see these equations, if a cell has a lot of ATP, it has a lot of energy? Would you say this is true? Who thinks it's true? A cell has a lot of ATP, does it have a lot of energy? Who thinks it's not true? Who thinks I need more information? Yeah. All right. Of course I need more information, because what I just told you is that the energetics really lies in this ratio of ATP to ADP. So if I want to know how much energy a cell has, I need to know the ADP concentration and the PI concentration, right? So really, the energy doesn't lie in the amount. A cell can have massive amounts of ATP, but it doesn't mean it has a lot of energy, right? If it has equally the same amount of ADP, it does not have more energy, right? So you really have to think about, where does biological energy lie? It lies in ratios, right? It lies really in this ratio of ATP to ADP, and this is what we call the charge ratio. And this is an important point. A lot of biologists get this wrong. They say a cell has a lot of ATP, it must have a lot of energy. This is not true.
I wanted to know, like, how, like, suppose if we know that ATP to ADP ratio is like 10, right? So how is this ratio maintained? As if ATP, this hydrolysis, is that a spontaneous reaction? And why does ATP, how is it stable inside cell? Why does it not get hydrolyzed?
No, exactly, right? Like, it's in continuous flux, right? This is what it's at steady state. So we assume it's at steady state. So meaning that the production rate equals the consumption. And there's a lot of research. How do you couple? How does a cell know how much it consumes and how much I need to produce? And this is a little bit of a control theory. Is it the demand which dictates the supply? Or is it the supply dictating the demand? Or to what extent can I be regulated? And there's a lot of intricate regulation, and all these feedback loops, this allosteric regulation we had yesterday will help you, right? If at one point the energy charge ratio goes up, there is this enzyme called phosphofructokinase, which does this first step in glycolysis and phosphorylating your fructose to fructose 1,6 bisphosphate. It doesn't regulate it by ATP-ADP ratio, right? And there are other enzymes in the cell which are really sensitive to this ratio, such as the AMP kinase, and that can then sense these fluctuations and try to help to compensate. And it will fluctuate on time scales of milliseconds. You will always have fluctuations, but on time scales longer than that, you can assume the steady state assumption. So it's really like the cell really is trying to keep this ratio. Oh, there was another question. Normally, it's a simplification. Normally, you would add the concentration of inorganic phosphate. And it is in here, sorry. Up there, PI. This is the inorganic phosphate. On the regulation, yeah, there is, right? Like, we have transporters transporting inorganic phosphate from the environment. And there's a lot of also cells are very sensitive to, you can starve cells of phosphate, for example, and they will. And we have also stores of inorganic phosphate, these polyphosphates in cells. Try to buffer this a little bit. I think there are pumps. Yeah, there are pumps. They're transporters. I don't know. Are there active transporters? Yes, there must be active transporters because it's a charged molecule. So you need ATP. I think there was another question.
I just didn't understand why it's the ratio that's important. So if the cell has lots of ATP, surely it has lots of potential to give phosphate groups, and isn't that a lot of, doesn't that give it a lot?
No, but if you, of course, you can do it initially if you have no ATP, no ADP around, but it's going to be so fast. But in the end, if you look at the equations, right, like the energy you're going to get is always going to be in a ratio, right? So for example, also the delta G0 up here is a function of the equilibrium constant, right? It's always a ratio.
So is ADP required for ATP to give its phosphate groups as energy?
Like if you throw just pure ATP in water, it's going to hydrolyze to ADP, and at one point you're going to have a little bit of ADP around, right? I think there's always going to be an equilibrium, right? There's always ATP, always going to be an equilibrium in water with ADP.
I still don't really understand. So if the cell is full of ATP, surely that has
But there's always ADP around, right?
Right.
So it's always in this ratio of ATP to ADP. Maybe I'm not getting the question fully. So in a hypothetical scenario where you have water and just ATP.
Yeah. Can the ATP not be used as energy? In that
The ADP can be used as energy as well, right?
ADP can be as well.
You have a constant flux, like at equilibrium, the forward and reverse rates are the same. So you have there's no net gain. Maybe we can talk about this later, right? Like, and hopefully to clarify.
So if we think about it this way, that the ratio of ATP to ADP, that sort of is a metabolic stress to the cell. If it drops, suppose it's a metabolic stress to the cell. But this metabolism is happening in minutes, right? The time scale of this is suppose in minutes.
Seconds.
Okay, in seconds. Now, if this metabolic stress is to activate a particular protein in order to, start a signaling cascade, and then, so what's the time scale of that? Is it?
So of course you have all this allosteric feedback, which we had like yesterday, and that also happens on these similar time scales. Otherwise, metabolism wouldn't work, right? But of course you have longer time scales where you involve transcription and translation. This is just like more of a long-term response. So you have a short-term response where you have enzymes which sense these fluctuations in these ratios, and these ratios also going to allosterically activate or inhibit other parts in the metabolic pathway. So you can have a short-term response trying to deal with this stress. Mm-hmm. And then if the stress continues for a longer period of time, you're going to have a long-term response trying to deal with it.
Right. So the time scale is sort of correlated to each other. Like something is in minutes, something is in seconds. But if they know that there is a requirement, they'll go ahead with the transcription, translation, and the protein will be.
Exactly. They coupled each other, right?
Right.
Yeah.
Mm-hmm. Mm-hmm.
Yeah. Exactly. So it happens that you activate and then you do the short term and the long term. Exactly. These are just, I think the question I was just asked, what is the ATP concentration, ADP concentration in different kind of systems? And you can already see that ATP is roughly in the lower millimolar range, right, from 1 to 10 millimolar. And then ADP concentrations is roughly in this high micromolar range. And then this is the concentrations of PI, and it's also the low millimolar range. And you can see this is roughly the physiological concentration, like physiological delta Gs you can harness from an ATP hydrolysis step. And it's roughly about 20 kT. But now, I just told you, is that a cell tries to maintain a high ATP to ADP ratio, which is required for life. But then glycolysis or metabolism makes ATP, so it has to fight that high energy ratio because it is much more harder. It's harder to make that ATP if you have a high ratio, which you need in order to sustain. And we already learned yesterday about glycolysis, and there are actually two reactions, and this is the beauty about metabolic pathways such as glycolysis, is the chemistry of these pathways design steps in these sequential enzymatic steps where you have a free energy change which is larger than the energy you need in order to synthesize ATP despite this high ratio of ATP to ADP, right? So this high ratio of high ADP to ADP in cells gives you a lot of free energy, but you also, if you have a high ratio like this, requires a lot of energy to make it. So the beauty about the design principles of metabolic pathways is that they make steps in these sequential oxidation steps, which then allows you to synthesize ATP despite this high ratio. Right? And on the other hand, we also have compartments in cells, such as mitochondria, which can, for example, change this ratio of ATP to ADP and make it cheaper to synthesize ATP. For example, the ratio of ATP to ADP in mitochondria is roughly 1 instead of 10 in the cytoplasm. Right? And there's two beautiful reactions in glycolysis, which exactly generate the deltas in these steps, which are larger than the hydrolysis, like larger than the energy required to ATP. And one thing we already yesterday went into glycolysis. One thing is this step in the lower glycolysis, which uses this molecule 1,3-bisphosphoglycerate. It looks like this, right? Like you have these two phosphate groups, and you can use one of those to transfer it to ATP, and the delta G0 is spontaneous. Right? It's about 4.5 kilocalories per mole. And it's interesting, the second enzyme, which was called pyruvate kinase, and it uses this molecule phosphoenol pyruvate and can use this reaction in order to phosphorylate ATP against this high ATP to ADP ratio. And this reaction, just without making ATP, has roughly about 15 kilocalories, or roughly times 4 in kilojoules per mole. And this is one of the most spontaneous reactions in cellular metabolism. And if you now couple this to an equilibrium to the synthesis of ATP, you can now really drive this reaction. These are just to highlight these two reactions which can synthesize ATP despite this high ATP to ADP ratio. There's only one problem, is what we learned yesterday. Glucose, the molecule we're going to use, doesn't have a phosphate. Right? And we know that putting a phosphate on without using ATP is not spontaneous. Right? So we invested 2 ATPs in the beginning to put 2 phosphates on. But however, we end up, and we use 2 ATPs, but however, we end up with 4 ATPs. So the net gain is 2, but we have 4 phosphates against 2 we put in. Right? So how do we gonna get an inorganic phosphate? We need another phosphate in this whole system in order to have a net gain of ATP in glycolysis. And we need to do this without using ATP, because otherwise we cannot do this. But we learned that making ATP or making a phosphate is thermodynamically unfavorable. So there is another really, really important concept in cellular metabolism, how we can harness energy without involving glucose, right? And this is an enzyme which is called, we already had it yesterday, was this famous loading control on your SDS gels, which is this enzyme called GAPDH, or glycerol aldehyde 3-phosphate dehydrogenase. And you can already see that, this is this molecule over here. And eventually we're going to end up with another phosphate up here. And it uses inorganic phosphate in order to make this molecule later on, which we can use in the next step to synthesize ATP. But if you write this down, you can already see that this is slightly unfavorable even at equilibrium conditions. So it actually needs energy, but under the conditions in cells, what I told you is always depends on the physiological concentrations of your molecules. This reaction is actually favorable. But how do we going to get the additional energy required to put this phosphate on this molecule? And there is something really interesting at this business end of this molecule, which maybe gives you an idea. I don't know how much chemistry you had, but this is an aldehyde group. And this one where the phosphate is an ester, but this one is actually carboxylic acid. So there's something happening here to this molecule before this phosphate is going to put. And this is what we call an oxidation. There is this classical view of we can oxidize molecules, For example, if we take an alcohol and your sugar is a polyalcohol, and we can oxidize this alcohol group up to an aldehyde and a carboxylic acid group. So what this currently did is we went from this aldehyde over here to carboxylic acid. And this involves the transfer of electrons. We take electrons away, and this is what we call classical oxidation, taking electrons away from this molecule. For example, going from this aldehyde to an acid group and oxidizing it is taking electrons away. And then the other way we can go is reduction, gaining electrons. So there's this nice brain teaser, it's called an oil rig, meaning that oxidation is loss and reduction is gain of electrons. And if you think about cellular metabolism, what catabolism is doing, burning wood, burning glucose, is nothing different than a chemical oxidation reaction. And similarly, our catabolism, or the respiration or the burning of sugar, is also an oxidation reaction. We go from this polyalcohol of glucose down to this carbon dioxide, the final oxidized form of carbon. And we're going to rip out electrons of that molecule. And this is the idea of catabolism, but we also sometimes want to build up, for example, fatty acids, which are completely reduced molecules. So basically, we have to reduce certain molecules, and this is kind of anabolism. So this is, if you think about the release of energy, and this is storage of energy. Of course, metabolism is not nuclear physics, so these electrons have to go somewhere, right? And this is where other energy currencies of the cell become really important, not only ATP, but we have a molecule which is called NAD or NADP, which can take up electrons to form NADH or NADPH. And this is called nicotine adenine dinucleotides, right? And the business end of this molecule, of course, electrons are not free in biology, and it comes in the form of this hydride ion, and it carries an additional electron that is charged. And that can go to this molecule of NAD+, and this hydride ion can be used, or these electrons from an oxidation reaction being reduced to NADH. So this molecule is an electron carrier in biology. So in this step of glycolysis, where we're going to get this inorganic phosphate in this molecule, we use the energy of the oxidation of this aldehyde group to an carboxylic acid group, while transferring these electrons to this electron carrier molecule, NADH, and forming NAD+, and forming NADH. And then this is oxidized, and then while this other one is. And there are way more electron carriers in biology. This is not the only one, but it's the most prevalent one. We also have other electron carriers. We have, for example, iron sulfur clusters in proteins, which can be oxidized and reduced and carry electrons. And so in the end, we have generated this molecule. And there's another really important point is, what are we going to do with these electrons? Right? They need to go somewhere, because eventually we're going to want to keep balance. Right? So what do cells do with electrons? Any idea? Exactly. So electrons you can use, right? You can charge a battery with an electron, right? With electrons. And this is exactly what biology can do with additional electrons. This is how we can harness that additional electrons. We can charge a battery. And this is where oxygen becomes very, very, very useful to cells, because oxygen really likes electrons. Right? So oxygen is what we call a really efficient electron acceptor. And it likes to take up 4 electrons and 4 protons to form 2 water. So you can reduce oxygen, right? Reduction is gain of electrons to reduce oxygen to water. And this is very useful for cells, because if we transfer these electrons from these electron carriers to oxygen, there's a difference in potential, and we can use this potential to charge up a biological battery, which you then can use again to generate. However, what is the problem if we don't have oxygen around? What are you going to then do with these electrons? You guys sometimes drink a beer or wine, something else like that? Never? Never. All right. Have you heard about Louis Pasteur? Right. And the Pasteur effect? Right. So this is the beautiful way how you can get rid of your electrons in making alcohol or making ethanol. And this is the Pasteur effect. This is the yeast cell making alcohol. So you have your pyruvate, and you made an additional NADH, which you carries electrons you need to get rid of in the absence of oxygen. So you can use a chemical pathway. So you have this aldehyde group, so you decarboxylate pyruvate, and you have this aldehyde group, which you now can reduce, so gain of electrons, and this aldehyde group reduced to an alcohol or ethanol, and kick this ethanol out of the cell. So you kicked out your electron, which you used, and you kicked it out of the cell in the absence of oxygen, and you made alcohol. And this is what a yeast does in the absence of oxygen to keep the redox balance, or the balance of your electrons. And what we do is when your muscle is anaerobic and we don't have oxygen around, you can reduce this pyruvate to lactic acid, which you can use these electrons from NADH. And I think I'm going to stop here. And this is fermentation actually regenerates your NAD plus and gets rid of your electrons.
Thanks a lot. We have time for a few more questions. One question over there.
So not really a question, just a comment to what he was asking. So I think like even if you have a lot of ATP, if let's say your corresponding ADP concentrations are also high, you're basically reducing the amount of energy you get, the free energy that you get from hydrolyzing the ATP, right?
Yeah.
So I think that is exactly the reason why the ratio would matter.
This is why the ratio was matter. Right?
Yeah.
Like the free energy is, well, the thermodynamics are always linked to ratios rather than the absolute.
Yeah, like you have the ADP too high, so like even if you have a lot of ATP, it doesn't provide you.
No, it doesn't
enough.
provide you more energy, right? Like
Yeah.
that's the reason why I ask you, like a cell, if it has a lot of energy, you need to know the ADP concentration, PI concentration to tell whether it has more or not.
So I had a question. So these transcription and translation rates, generally they won't be the same for, let's say, 2 different genes or 2 different mRNAs, depending on several different regulatory factors.
Roughly, roughly
So
the same.
is it
similar.
useful to think about averages like this? Because
yeah, so like of course you can have obstacles in your way, right? But normally you have other enzymes removing this obstacle. And these transcription and translation rates are roughly like a good approximation of the average rate.
So like I had a question about what happens with these free electrons.
Yeah.
So they either react with oxygen or they make ethanol out of pyruvate, right? So are these
in the yeast they make ethanol, in us they make lactate, yes.
Yeah. So are these, I mean, do these steps actually kind of help with, I mean, they are high energy steps, so you would get a lot of energy out of them, like especially when you kind of put oxygen and 4 electrons, right? So is this energy used or is it just more like to quench the reaction?
No, like this is of course used, and this is the principle of oxidative phosphorylation or chemoosmosis in your mitochondria. And if I would have enough time, I would have a few slides, and maybe we can start with that in the lecture, just to say you can use these electrons to charge up a biological battery and then use this energy to make ATP.
Sir, given the genes are unregulated, will their mRNA degrade?
I didn't hear the first part of the question, sorry.
Given the genes are unregulated,
Unregulated.
will their mRNA degrade?
Yeah, like RNA is unstable, right, in a way. I don't know what the half-life time of a pure RNA molecule in water is. Someone can help me. Does anyone know? But it will degrade eventually. It will hydrolyze because you have if you think about the chemistry of you have your additional OH group, which usually will nucleophilic attack somehow eventually, though it will degrade for sure. I don't know the half-life time in water.
Other questions? If not, we can thank Jonathan again. And so now we have a coffee break and we'll reconvene at 11:30. Thanks.